Hi, this is Dr. Aristo Vojdani, an immunologist with more than 50 years of experience in research and clinical immunology laboratories. I am Chief Scientific Advisor to Cyrex Laboratories and Technical Director of Immunosciences Lab, where I developed more than 300 different immunological assays for early detection and hopefully for prevention of autoimmune diseases. The science behind some of these tests could be summarized as: our genome can affect our exposome. Our exposome can affect our microbiome. Our microbiome can affect our immunome, and our immunome can affect our diseasome. On Saturday, April 17, the coming Saturday, I have the honor to speak on behalf of Cyrex at A4M MMI meeting on the subject of environment, neuroinflammation, and autoimmunity. So in this meeting, not only I'm going to talk about performance characteristics of laboratory testing, but I'm going to talk about mechanisms by which food, toxic chemicals, and pathogens affect our gut microbiome. The gut microbiome will release a variety of toxins that can damage the gut barriers, and when the gut barriers are open, many unwanted macromolecules can get into the circulation, where immune system reaction against them results in variety of antibody production. These antibodies, plus other mediators such as cytokines, plus lymphocytes such as T helper 1, T helper 17, all together can damage the blood-brain barrier, where inflammation in the brain results in neurodegeneration and neuroautoimmunities. The good news is that these antibodies could be detected 5 to 10 years before the full-blown of autoimmune diseases. So these antibodies, the meaning antibodies produced against food, toxic chemicals, pathogens, tight junctions, and many other tissues in our body, could be detected 5 to 10 years before the onset of autoimmune diseases. That's why we call them predictive antibodies. And so there is a window of opportunity for clinicians to intervene and prevent development of autoimmune diseases. Also earlier, I mentioned that our exposome affects our immunome. Fortunately, a new test which I developed for Cyrex will be available very soon, which is called lymphocyte immunophenotyping. This is not just about T cells and B cells and CD4, CD8 and their ratio, but Th1, Th2, T helper 17, regulatory T cell, and variety of natural killer cells. So by combining the predictive antibodies plus lymphocyte immunophenotyping, clinicians, practitioners can find the early events in immune dysregulation and autoimmunities. So by intervention can stop or even reverse the course of autoimmune and many other inflammatory diseases that affect between 10 to 20% of the world population. So I hope to see you on Saturday between the hours of 11:45 to 12:45, and I'll be very happy to see you there and give you more detail about the subject of environment, neuroinflammation, and autoimmunity. Thank you.